వాట్సాప్ నాకు మెయిల్లో లేదండి. ఫస్ట్ లేదు మ్యామ్, నాది పర్సనల్ మెయిల్ పెట్టాను ఆ పైన మెయిల్ మ్యామ్, అదే ఆ పైన.
Students, an email ID is displayed on the projector. Send your posters to the email ID which is given here.
Did you get it? Shape was right.
Yeah, I got it. This one. Yes. Open it. Yeah, your time starts now. 5 minutes from now.
Good afternoon, everyone.
Mail ID.
Myself Sheikh Vishwara. I'm from MSc first year Microbiology of our college. So I'm just going to give a brief presentation on probiotics. So basically, what is probiotics? Probiotics is a living organism which is present in foods and supplement, which helps to maintain our gut system with the help of good bacteria. So here are few characteristics of probiotics. As we all know, probiotics is very important for our daily life. It's very important to help our intestine for digestion and all. So it helps to maintain the low pH in our intestine with the help of lactic acid. It also produces beta-D galactose enzyme, which is the breakdown of lactose, and it produces certain vitamins which are really essential for our intestine, like vitamin K and B. It is also anti-inflammatory, anti-mutagenic, and it is non-toxic for our body. As we all know, probiotic has both advantages and disadvantages. If we have effects of probiotics, then we must suffer with constipation and I will hand over the mic to my partner so that she gives you more about probiotics.
Good afternoon everyone, myself is Shravanti, I am from MSc Microbiology of Plants, Vivekananda College. I am going to continue the remaining part of the presentation, which is probiotics and disease prevention and treatment. As we know that probiotics helps us to maintain and decrease the prevention of certain diseases like colon cancer, which is where the probiotics like Lactobacillus bulgarius helps us preventing the breakdown of the enzyme called beta-glucuronidase that contributes to the growth of the cancer-causing agents. And the another one is the probiotics helps us to decrease the lowering the cholesterol level, where the breakdown of the bile happens in the gut with the help of the lactic acid bacteria. And another one is reduction of the blood pressure and antibiotic associated diseases which causes by the imbalance of the microbiota in our body. Some of the treat allergies like which helps in degradation of antigens and normalization of the properties of the microbiota in our body, and regulation of the anti-inflammatory mediators helps in stimulating and response of the immune system. And some of the probiotic strains which we can mainly see like common examples are Lactobacillus species like Aspergillus and Plantarum, Bifidobacterium species like Bifidium and Longum, and some of the other species like Streptococcus thermogenes. Thank you.
Thank you for completing the time. We request the audience to maintain silence. We can't hear the presentation properly. Please cooperate. I request all to maintain silence.
Nazmeen and Saujanya, please present your work.
This one, right? Yes. The order. Your Soumya, Saujanya and Nazmeen. Nazmeen Tabassum. Yeah, I will share. Can you please? We are not holding it in order. Google is already registered. It is very user.
Yeah, your time starts now. But now 5 minutes.
Good afternoon to everyone present here. Me, myself, Nazmeen and Saujanya here, we are there to present the microbiome of marine. So as we all know that marine is the largest ecosystem, not only as part of aquatic animals but also for our microorganisms. One single drop of saline water contains about 1 million of microorganisms and they account for about 98% of total biomass of the entire ocean. So in our poster we have briefly explained about the products, genomic products which we are getting through the marine microorganisms. As microorganisms are many in the marine system, we have many applications and uses of them. So, first of all, they act as photoprotective anti-aging agents, which is a very crucial product in the cosmeceutical industries nowadays. Also, they act as bioemulsifiers, bioreluctants. They are used in many industries like dairy, detergents, textiles, and many applications as well. Saujanya will tell you about the derived compounds.
Good afternoon, everyone. Myself, Saujanya.
Little mic.
Marine derived components, some of them are glycerins. Glycerins is a cosmetic product isolated from fluorocarbons which act as anti-inflammatory agent and process analgesic activities in humans. Absine is a cosmeceutical product composed of mainly exopolysaccharides and produced by deep sea algomonas. Ionelix is developed by Pharma company and it is isolated from sea squid, used to treat soft tissue sarcoma and ovarian cancers.
Yeah. Alankrita and Sri Harshita.
I said no talking means no talking. That's it. Yeah, 5 minutes from now.
Good afternoon, one and all. I'm Alankrita, Department of Microbiology, first year. And I'm Sri Harshita from Department of Microbiology, first year. So, first of all, everybody know about microbiome. We have been discussing about plant and animal microbiomes. But we know that there is another microbiome that is residing on our skin, that is skin microbiome. A topic which is very less explored. So our today's article presentation would be skin microbiome, current landscape and future opportunity. So we all know what is a microbiome. Microbiome is a group of community of organisms residing at a given habitat. So if it is skin microbiome, that means microbiome present on the skin. So Harshita will give you the objectives about this paper.
So basically, the microorganisms living on our skin will be our biggest asset, because as humans, our skin is the largest sense organ that we have. So when we use skincare products that many of you will be using, I guess, so they should match our skin's pH, sebaceous gland density, and humidity and temperature, because a slight difference in these biological characteristics can cause a lot of damage to our skin microbiome. Because we have studied in our microbiology labs that if we increase the temperature, this particular organism will grow, or if we decrease the temperature, this particular organism will grow, and also the pH changes infecting the bacterial growth. In the same way, our microorganisms on the skin will also act accordingly to the biological conditions that we provide to them. So it is very important to take care of this particular aspect of our skin, because once we break this balance, the skin will start showing effects like rosacea, acne breakout, and dryness of skin. And methods will be given by Anushka.
Now there are various methods which are highly evolved using a lot of technology for this skin microbiome. So these include the next generation sequencing, metagenomic technologies, metagenomic annotation, metagenomic analysis, and metagenomic enrichment.
So on a conclusion, what we want to present as an overall takeaway from this presentation is that we have to spread more light on this particular aspect of microbiome, because this is an area of research where we can get excellent results with very fine observations. Thank you so much.
P. Sirisha.
Which bill?
Yeah, this one.
No.
Not that.
Then I got the mail.
T. Divya, please ready to present your poster next.
Send it once.
I'll send it to this mail.
Okay.
What happened? I'll send it now.
From which mail you have sent, huh?
Search them.
To him on which mail?
Mail.
Side is people's mail.
Your email ID.
But I
sir.
got from this.
Not that.
Sirisha.
Not that. Yeah.
Where is your email
Divya, please present your
ID?
poster.
Divya's poster is ready.
From this one, right?
Divya.
Only order is same. You get your chance.
And this one is the one.
Yeah, 5 minutes from now.
Yeah. Ma'am, ఒక్క నిమిషం.
Divya, 5 minutes from now.
It's there. It's scrolling is also there. Wait, I will see. Because okay. Okay. I will do it.
Good afternoon, everyone. I am Divya, MSc second year student of Bhavans Vivekananda College. I am going to present on the topic harnessing bacteriocins modulating the gut microbiota for optimal health. Our human gut microbiota contains trillions of bacteria, fungi, and yeast. Any imbalance could cause diseases such as IBD, diabetes, obesity. And to treat such diseases, we use antibiotics. And antibiotics usage is significantly increased, which not only disturbs the composition and function of the gut, but also encourages the selection of MDR strains. So bacteriocins are, to address this problem, we can consider bacteriocins. Bacteriocins are small peptides, antimicrobial peptides that are produced by lab
lactic acid bacteria, and they are heat stable and they are active against wide pH range. And objective of the study is to emphasize the link between bacteriocins and the human gut, and also investigate the role of bacteriocins in shaping the host microbiota, and also to highlight the potential therapeutic activities of bacteriocins. Purified lysine, which is produced from Lactococcus lactis, it depletes the Clostridium difficile without affecting any native gut bacteria, and also the bacteriocin produced by Lactobacillus crustorum is active against E. coli, S. typhi and S. aureus. And enterocockin B also inhibits Staphylococcus, Clostridium and other pathogens. This picture summarizes the potential functions of bacteriocins. It is not clear, but it is involved in anti-obesity activity. It will regulate the gut-brain axis, and it will also modulate the immune system by increasing the level of CD4 and NK cells, and also involved in apoptosis of cancer cells. And bacteriocins will act against both gram-positive and gram-negative cells by pore formation, and it will also inhibit the translocation and migration of bacteriocins to extraintestinal organs such as liver, kidney and lungs. This picture is a summary of mode of action of bacteriocins that are discovered till date. Lysine, listostaphin, spidiocin. These are all the bacteriocins which are produced from gram-positive bacteria against gram-positive bacteria, and they involve in pore formation and inhibition of peptidoglycan synthesis. And in gram-negative cells, their action is by ion leakage and pore formation. They also inhibit by direct contact. And future prospects are that bacteriocins can be considered as next generation antibiotics, therapeutics, because they are also able to target the multiple resistant strains. I want to conclude saying that bacteriocins participate in the modulation of gut bacteria by their safety and better efficacy makes them superior to existing therapeutic agents. So it is necessary to continue performing further exploration, development and utilization of bacteriocins. Thank you.
Are Sirisha, Sushmita and Srivarsha.
Good afternoon, everyone. Myself T. Sirisha. I am in the Department of Microbiology. I did a project research project in the bioinformatics and computational microbiology under the guidance of Dr. Rajashekhar Chikati. He is from the Department of Biochemistry in the Usmania University, and this is my abstract which have been done. Basically, it refers to the science which is dealing with some of the databases and the biological databases, and these are the targets of the sites, and which have taken as docking studies of the modeling, that is from TCR. It is an Homo sapiens which is developed from one of the PCR technique, and it is of one of the isoform. And introduction, basically it is an interdisciplinary science. It consists of three-dimensional things, which is risen from the many of the Homo sapiens and from the genomic studies. And aim and objectives is it is used to predict the 3D structures, and it is used to evaluate the 3D structures and build the 3D structures from the modular. And even by using the bioinformatical tools, I can build the 3D structure from the modular. And the methodology I do follow is from various bioinformatical tools such as FASTA, ProSet, and PDA, EMDL, and Ramachandran plot by the Savers method. These are the things which I followed in the methodology, and this is the result of BLAST. In BLAST, it consists of graphical summary, taxonomy, and descriptions, different types of assessments, and based upon the assessments, the multiple sequence alignment is present. And this is one of the cluster omega. It consists of phylogenetic tree, where it consists of bacterial species from one to the other, and even I have predicted the 3D model, that is PyMol, and it is an from the pathogenic interaction between the species. And this is the PD sum, which PDB sum. It is one of the secondary structure of TFC3, and it consists of Ramachandran plot. It is the servers where which have been isolated from the TCF factor. And coming to the summary, basically it is an pharmaceutical use. It is used for the drugs to differentiate between the gene and the protein structure. Not only to recover the protein, but also to build up the structure between the gene and the protein sequence. And some of the bioinformatical tools which have been used is, these are the bioinformatical tools, and the references which I have taken from the research papers such as evolution and the physics, and dynamics and the stimulations at the constant pressure. And this is my research project, and even I published the paper on this project. Thank you.
R. Sirisha, Sushmita and Srivarsha.
Good afternoon, everyone. I am Sushmita, and my team members are R. Sirisha and G. Srivarsha, and we are from Bhavan's Vivekananda College, pursuing MSc second year microbiology, and our poster represents about probiotics in the battle against cancer. Gut microbiota plays an important role in the human health. Sometimes it can be lost by the factors like antibiotics, stress, and environmental sources. So it is important to restore this gut microbiota by maintaining a balanced diet. So probiotics plays a major role in restoring the gut microbiota. Some of the main examples of probiotics are Lactobacillus, Bifidobacterium, and Lactococcus, Streptococcus thermophilus, and Propionibacterium. So probiotics, the main characteristics of probiotics are, so the probiotics are non-pathogenic because it does not cause any harm to the body, and it can tolerate the bile salts, and it is mutualistic associated with host, and probiotics survives the gastric juices, and it is regarded as safe to consume. From your Siddhesh will take on.
One of the most crucial characteristic of probiotic is treating the cancer. So, probiotics has played a key role in preventing cancer. So, nowadays it has become a very primary health hazard. So, you will be wondering how the probiotics will treat the cancer and their mechanisms. So, there are different mechanisms which are followed by the probiotics to prevent from the cancer. So, the first one is down regulation of the oncogenes. These oncogenes are these are the genes which promotes the cancer. So when we consume the probiotics, it reduces the expression of these genes. And the other mechanism is apoptosis. It is referred as a programmed cell death. It interacts with the immune system and modulates the signaling pathways. Then kinase inhibition. Kinases are the enzymes which are important in our body for
cell growth and division. So when we consume the probiotics, these probiotics will reduce the kinases and it doesn't allow the kinases to multiply the cells. And it also inhibits the mutagens. Mutagens are the chemical agents or viruses which causes the cancer. These mutagens can be inhibited by the probiotics, and it also acts as an anti-mutagenic agent. So there are different mechanisms which are performed by the probiotics to prevent the cancer. So the probiotics that can prevent the cancer are of different types, such as colorectal, liver, breast, and bladder, colon, and cervical. And these are the tabular representations and also results of the clinical trials where they found that these probiotics can treat the cancer and also different strains which helps to treat the cancer. Not only eating fermented food, but also other measures are to be taken, such as supplements for those who are lactose intolerant, and prioritizing fiber-rich food, and managing our stress as a gut-brain barrier. And here are some graphical representation for normal food and probiotics. And here is the graph for
Your time is up.
here we can conclude that research is being going on, treatment is ongoing. Current evidence is not still we end our topic by saying a small quote: "Gut love, cancer shall."
Small announcement. BSc students whose posters or abstracts are not published in the physical copy of Sovereigns and who have submitted late, be ready, submit your abstract or posters to Deepak's mail ID. Next.
PPT నాది
Yes. Mic. Ravish, mic. 5 minutes, once you start. Yes. Yeah, 5 minutes from now.
Good afternoon to everyone. I respected dignitaries and panelists present here. My name is Rajesh. With my teammates, we are going to present you a review-based e-poster on microbiome and its various aspects in the relationship between human health, mental health, and sustainable agriculture. As in our poster entitled as a harmony in diversity, microbiome is one such topic which interlink to various aspects of our lives. So our poster is a review-based poster. We are divided the topics into its impacts on our human health, our mental health, and how it helps in sustainable agriculture. As of now, in human health, from this picture you can see we have microbiomes on skin, on gut microbiota. So all over the world, we have now completed around 130,000 genomics of our microbiota, of which 30,000 genomics have been done. So these are the data which helps in maintaining our microbiota. Now my friend Narendra will carry on with sustainable agriculture microbiomes. Our microbiomes which are present or which helps in agriculture.
Mainly, microbiomes are helpful for the crucial role in the sustainable agriculture. Mainly, these are bacteria like nitrogen-fixing bacteria and digestion-fixing bacteria. Such as examples are Bacillus and Rhizobium. They help in the nutrient management and plant growth development. by fertilizer developments.
I'm going to be speaking about the psychobiotics. Psychobiotics is one of the new area of the study. Mainly it deals with the interaction. Mainly this field explores about the connection between the gut microbiota and also our mental health as well. And it also given the presence of some factors like antibiotics and some diet and also some various environmental factors like alcohol consumptions and stress responses as well. So as we compare the diets of our ancestors and our in the modern era of the diet, there is a completely difference are there. And upon and so the uptake of this diet and the antibiotics usage, it leads to the changes of these microbiomes in our gut. So due to this, we can able to differentiate between the microbiomes of our ancestors and also the modern microbiome. And also, some researchers have also tried to explore the new class of the interventions which are called as the psychobiotics, and which especially aims to reduce the levels of the anxiety and as well as the depression, and also one of the antidepressant solutions, and also the anti-inflammatory responses as well. And also they have also tried to manipulate the this gut microbiota by introducing some kind of the probiotics and the prebiotics, and as well as the fecal transplants as well. So upon this introduction, so it also resolve the anxiety and as well as the depression problems. So in the conclusion part, so these recent advancements of genomics and also the next generation sequencing, it also it gives a lot of aspects of finding a relationship between this microbiotics which are used in the basis, and some of the epidemiologists also.
Your time is up.
B. Kaveri and S. Divya.
Good afternoon, everyone.
From now, time starts for 5 minutes. Start.
Good afternoon, everyone. My name is Divya. I'm my friend Kaveri. We are from MSc Microbiology final year of Bhavan. The topic of our poster is about the prebiotics as health booster. Nowadays, antibiotics are consumed frequently. So frequent consumption of antibiotics, processed diets, and stress are disturbing the balance of our gut, leading to health issues. So the solution for this is by incorporating the prebiotics and probiotics into our diet can restore the balance of our gut. Prebiotics was given by Gibson and Robert Roy in 1995. As we all know, prebiotics are the non-digestible fibers, which are the short chain of sugars between 2 to 10 sugar units. Prebiotics are not digested by our body, but the prebiotic fibers act as a food for good bacteria such as the Bifidobacterium and Lactobacillus. The prebiotic foods are the apples, oats, artichokes. These foods not only add a flavor to the food but also provide a healthy dose of prebiotics. Common prebiotics are fructooligosaccharides, galactooligosaccharides, inulin,
polydextrose, etc. Inulin travels in the colon and becomes a new source of bacteria such as Bifidobacterium and Lactobacillus. These bacteria selectively ferment the inulin to produce short-chain fatty acids such as lactic acid, butyric acid, and propionic acid. For a food to be considered as stomach acid, enzymes, or digestion, it should pass safely to the large intestine through stomach and the small intestine. In the large intestine, the objectives include prebiotic sources and health benefits, direct and indirect effects of prebiotics on the gut epithelium and immune cells, the effects of prebiotics on various human organs.
Good afternoon, everyone. I am going to explain about methods. Methods include both the preclinical and clinical trials according to FDA. Preclinical trials include both in vivo and in vitro studies, and clinical trials include 4 different phases. In phase 1, the prebiotics are tested for their safety, and in phase 2, they are tested for their efficacy. Phase 3, they are tested against stability, and in phase 4 they are tested long-term run and what are the advantages and disadvantages of the long-term run. In the second picture, we
అయితే
can
ఒకసారి
observe
వస్తావా నా
that
ల్యాప్టాప్ లో రిపోర్ట్ లో ఇంకొన్ని ఇన్సర్షన్స్ చేయాలి వచ్చి చేసేవా అక్కడ
that are both direct and indirect. Direct mechanism include production of IgA
పిల్లలు ఎవరు కనిపించు
and also epithelial
మహేష్ ఏడి
lining. Indirect effects include production of enhancement production of more short chain fatty acids, and also help in promoting the growth of probiotics. And according to literature, prebiotics have many different effects on different types of organs. Like in brain, they help in preventing dementia, and also help in learning ability, and also we can remember. And in lungs, they help in prevention of respiratory tract infection. And in heart, they help in reducing cholesterol levels, therefore reducing the number of cardiac arrest patients. And in gut, I conclude that prebiotics are also helpful. Thank you.
Anusha and group, PSC.
What will be the name? It is one.
They are not there in the shop. 1 second, Amma. They write it.
It's a PPT. It's a PPT, right?
Yeah.
I will share.
1 minute. Sir will write the title and then I say start. Then
This one, okay?
Monica, sir. Monica. Yeah, you can start.
చార్జర్ ఉందా
Firstly, I would like to thank Bhagavan for giving us this exposure, and I would like to thank Mrs. Chaitanya for being our constant support in the entire research. I would like to thank Mrs. Chaitanya for being our constant support in the entire research. Our topic for today is utilization of agri-waste, that is groundnut shell, rice bagasse, pineapple leaves for preparation of paper. Synthesis of nanoparticles and their applications. This is the original research done in the laboratory by conditions.
Only participation.
The objective of this project is to promote use of agri-waste. We have taken groundnut shell, rice bagasse and grass bagasse, pineapple leaves as raw material. The pre-treatment is done and the pulp is obtained, to which different kinds of paper are obtained. We have used crafts method and soda pulp method for the production of the paper. We have used crafts method and soda pulp method for the production of the paper. We have observed that during crafts process we have used for groundnut shells, and we observed that the paper and rice bagasse paper was of good quality, and we obtained good results.
Good afternoon, everyone. I'll just detail about the experiment that we have carried out in laboratory conditions. That is, we've used 4 different agricultural waste products. It is groundnut shell, rice bagasse, grass bagasse, and pineapple leaves. All these 4 were easily available that were locally, and we have taken all this and we have generated a paper that is made using 2 different methods. One is craft process and one is soda pulp. The only difference between these 2 processes is use of 2 different chemicals. It was sodium hydroxide and sodium sulfide, and in soda pulping we've used washing soda, that is sodium carbonate. The only difference that we could make out after production of paper was the holding strength. The paper that was produced during soda pulping has given proper rigidity and proper strength to the product. After that, on the other hand, we have simultaneously worked on synthesizing silver nanoparticles that was also generated by onion peel. The synthesized nanoparticles, they have been extracted, and the product that we have developed from the paper, that was from rice bagasse, we coated it using 2 methods. One is spraying the mold, and the other one is making a thin coating. Then concluding that, in our experiment, which was completely done in a laboratory condition, we've used 2 vegetables. One is tomato, and the other one is a perishable food, that is green leafy vegetable. We've stored this simultaneously. We've observed it for continuous 7 days, day by day. The food that was stored in the product that we have made from rice bagasse was clearly not spoiled, and it was fresh even to the 7th day. But the food that was stored at room temperature has been spoiled on the 4th day, and now the perishable food that was green leafy vegetable that we have used as spinach, that has showed its spoiling nature on the 2nd day. So concluding, I'd like to say that even though in the laboratory condition we've used rice bagasse, this can be altered like with tissues. Thank you, ma'am.
Next. Harshan group. Next.
What's group in group? It will be tough to on the WhatsApp here. Which is
I mean, nearby
I related it not in the which microbiome.
You are not sent to the souvenir, right? New entry. What's your name? Is there?
Harsh Jain.
What's your title?
guardians of the
Understanding the influence of prebiotic carbohydrates on gut health? No? You know what's your title? Yeah. Oh, new last time. It's okay. Yeah. We have many other posters. 5 minutes from now.
A very good afternoon. This is Risika Kumari and this is Harsh. We are from FIC 3rd year. Our topic is guardians of oral health, prebiotic enhanced probiotic in the battle against Streptococcus mutans. As we know, the humans are natural. As we know, the humans are mutually and possibly associated with the microbiome. And probiotic is a component of a microbiome which mainly involved in the maintenance of gut health and oral health. And due to frequent consumption of carbohydrate, it causes alteration in the oral environment by creating a low pH. And this low pH environment favors the growth or facilitate the growth of the probiotic or bacteria, which are generally present in 2% in total biofilm. And the bacterial strain is Streptococcus mutans. And this Streptococcus excessive growth in turn leads to the dental demineralization and caries questions. And this poses a major threat to the oral health. And this study mainly explores about the prebiotics effects. Efficacy of this study mainly explores about the efficacy possessed by prebiotic on the enhancement of the probiotics. Probiotics are mainly beneficial microorganisms, and here predominantly was Lactobacillus acidophilus. Lactobacillus acidophilus inhibits the growth of the S mutants, which are generally present in the oral cavity, oral environment. This study shows when L. acidophilus and Streptococcus mutants were co-cultured together in MRS medium,
supplemented by with the prebiotics. There is FOS and GOS. FOS is fructooligosaccharides and GOS are glucooligosaccharides were supplemented in the very concentration of 2, 3 and 5%. There is a significant decrease in the S mutant growth rate. And from your graphs, we can see that the L-acidophilus and S mutants were co-cultured together. There was an inhibition of the S mutant bacterial strains. And your question.
Good afternoon to everyone. I am Harshin from MIC. Now I will continue conclusion. This study determined the growth rate of S mutants when the co-culture when lactobacillus acidophilus. The growth rate of L lactobacillus acidophilus were unexplained by the prebiotic fructose oligosaccharides and the GOS. The growth rate of the S mutants and unexplained by the prebiotic reduced by the 3% of FOS and 4 chemical and the mixed rate. The 1% of FOS and 3% of the GOS concentration appears to have the ability to slow down S mutations. Another study touched on the re-enhancement the membrane fatty acids.
Next, Nomita and Garima.
Start. From now 5 minutes.
Good afternoon, everyone. I am Garima from Bhavans Vivekananda College, and I'm pursuing my bachelor's in microbiology, genetics, and chemistry, and I'm a first year student. So today I'm very pleased to have you all here for my poster between plants and their microbial companions. So minutes the green canopy exploring the hidden world of plant microbes. What is the plant microbiome? The plant microbiome is the number of microorganisms which are present around and inside a plant tissue. And the ecosystem plays a crucial role in their overall health and well-being. Just like the ecosystem supports us to survive in this environment, the plants also receive this help from the nature and thus they are able to survive. So let's talk about what kind of help it is. What is the importance of the plant microbiome? Plant microbiome helps in nutrient cycling, which is breaking down of organic matter and then releasing out nutrients. Disease resistance. This microbiome also protects plants from pathogenic microorganisms by acting as a barrier between the pathogens and the plant tissues. Stress tolerance tolerates environmental stressors and environmental stressors like hypersalinity, high temperatures, and drought conditions. Now that we know the importance of them, let's find out what is the component of this microbiome. So this microbiome, you can majorly classify it into 3 parts. The first is the rhizosphere microbes, phyllosphere microbes, and endosphere microbes. So rhizosphere microbes is the region present beneath the soil. That is the amount of soil present around the plant roots. They compose the rhizosphere of that plant. And here you can have bacteria and fungi, both of them. They help in promoting the nutrient uptake for the plant. And if we talk about examples in fungi, they can be trichoderma and pseudomonas in bacteria. These have a symbiotic relationship, which we'll be talking about a little further. Then there is phyllosphere microscopes, and now phyllosphere comprises of the aerial parts of the plant, including the stem. Okay, now including the stem and the leaves, which protects against the pathogens. And then there is endosphere, which is endophytic bacteria is present inside the plant tissue to provide defense to it. Applications is it is used in biofertilizers and biological control and crop improvement. Its significance is to increase the yield and productivity. And can I just give the conclusion? Thank you.
We have others waiting, right?
Sure.
Nikki and Anchal.
Thank you. Give it.
Excuse me. What is the name?
आजकल करके आएगा आजकल.
The next one.
మేము ముగ్గురు ఉన్నాము కానీ వాళ్ళ పేరు ఒకరు కూడా పడింది నాగాంజలి ఇంకా
Yeah, start. Your time starts now.
Good afternoon to everybody present here. I'm Anshul Singh of MCC first year. My partner and I are here to present a poster on the topic Power of Probiotics. So what are probiotics? Probiotics are live microorganisms. To be specific, they are live bacteria and yeast that are good for you, especially your digestive system. Bacteria are all over our body, good and bad, but these probiotics refer to good bacteria or the health-friendly bacteria. Initially, probiotics were referred to as microorganisms that have effect on other microorganisms. The concept of probiotics involves the notion that substances secreted by one microorganism, they stimulate the growth of other microorganisms. Moving ahead, let us talk about the health benefits of probiotics. Let me talk in regards of anxiety and depression. Research shows that there is a relationship between our gut and the brain, a partnership which is known as the gut-brain axis. And the primary connection between the gut and the brain is the vagus nerve, which is indeed the longest nerve in the body. So what these probiotics do is that they help reduce our levels of stress and anxiety. They help us to elevate our mood and also enhance our cognitive impairment. For an example, a study published by the Frontiers in Aging Neuroscience showed that Alzheimer's patients who consumed milk containing 4 different probiotics scored better in a test which was conducted in order to measure their cognitive impairment than the Alzheimer's patients who consume a regular or normal milk. Another example is that women who had consumed yogurt containing a mix of probiotics twice a day for 4 weeks were calm when they were exposed to images of angry and frightened faces. This was in regards of anxiety and depression. Let us talk about gut health now. As we all know that there are millions of microbes present in our body, and the role of probiotics is to prevent and treat dysbiosis, which is a condition of imbalance of microbes in our gut microbiome. When these probiotics work, the good bacteria in our gut, they increase in number.
Last 1 minute.
Alright ma'am, now my partner will conclude.
There are various sources for probiotics like yogurt, kimchi, paneer, cheese. But before consuming any probiotics, we need to consult any practitioner or doctors so that we do not get disaffected or any disadvantage happen to us. So we need to consult doctors before taking probiotics and also taking care while buying the probiotics by reading the mentionings or the contents of the probiotic. Thank you.
Naganjali and
Take them. Last minute entry. Deepak Perunda.
Yes, ma'am, last.
Yeah. Deepak.
Let side only.
Yeah, from now 5 minutes.
Good afternoon to the respected dignitaries and honored friends. Today we are here to post a presentation on the topic microbiome associated with colorectal cancer. Till now we have seen that microbiome plays a very positive role either as symbiosis and commensalism, but we often forget them as determiners. So here the topic starts. Microbiome associated with colorectal cancer. Colorectal cancer is one of the most common type of cancer and it is the 3rd highest cancer causing death worldwide. Numerous studies has told us that this is somewhere related to the Western diet and dietary fiber. So we can come to the statement that it is related to the gut microbiome.
and how it influences the gut microbiome on colorectal cancer progression. It disrupts the dysbiosis. It disrupts the normal count of gut microbiome, which leads to the chronic inflammation. Here you can see there is chronic inflammation, which leads to the cancer progression. The metabolites is altered, short chain fatty acids and secondary bile acids are altered. Imbalance in the metabolites lead to the chronic inflammation. And we can see the correlation between CRC and gut microbiome. We have taken a certain people suffering from colorectal cancer and we have seen that they had lost the richness in the specific microbial community. They have not, and this shows a great impact on the mucosal immune response, which is not found in the healthy individual. And the total 11 OTUs are found, which include Streptococcus, Peptostreptococcus, Klebsiella, and etc. Influence on the gut microbe on colorectal cancer treatment. Now we will see the methods of treatment, which include chemotherapy, radiotherapy, immunotherapy. In chemotherapy, we can see the FMT told by the sirs also. In this chemotherapy, the administration of antibiotic, it reduces the efficiency of oxoplasmosin and CPS. And in radiotherapy, the pelvic areas get dysbiosis, which we can clearly see that it is the main impact of colorectal cancer. And immunotherapy is one of the major treatment which can be followed.
Continuing the rest of the further topic of this. Now the influence of the gut microbiome on CRC formation. There are two cells. One is carcinokin and chemokine. They both formed and produced by the cancerous cells, which lead to the myeloid cells and produce T cells. Now coming to the pro-tumorological microenvironment, characterized by the synthetic and synthesized and growth of the angiofactors. Now coming to the examples, E. coli and Bacterium, these both form in the large intestine, whereas they produce more common symptoms like diarrhea and ulcer problems. And coming to last two, Lactobacillus and Bifidobacterium, they are good eco-friendly bacteria which helps to fight against the bad organisms. Now coming to the modulation, it uses FMD treatment and probiotic therapy, in which FMD uses fecal microbiome transplantation. It includes transplantation fecal and from the healthy microbiome. Thank you.
Abhilaya and group.
Anyone else?
This one, right?
Presentation.
Yeah. From now 5 minutes.
Good afternoon to one and all present here. I am Abhinaya, this is my teammate Nikita, and we are from BSc second year Life Sciences from Pavan Vivekananda College. And we are presenting our poster on the gut-brain axis and the role of gut microbiota in neurological disorders and how prebiotics and probiotics are used to modulate the immune pathways. So before I even begin my presentation, I would like to ask everybody here, how many of you all know about Stephen Hawking? Please raise your hands. The famous physicist. Okay. So did you all know that he suffers from a neurological condition called ALS, that is amyotrophic lateral sclerosis? In this disorder, actually what happens is the nerve cells progressively degenerate and the person loses their voluntary control. However, the intelligence is not affected. I mean, clearly, Stephen Hawking, right? So, like that, in this paper, there are about 7 neurological conditions that are mentioned. And what they've seen is the people affected with these conditions have a different gut microbiome composition than the ones that are not affected. So how are we connecting these two entirely different systems? One is the gut and one is the brain. They're entirely different, right? How are we able to connect them? It is because of the gut-brain axis. And there are 3 things we can remember about the gut-brain axis. 1 is it is complex. 2 is it is bidirectional. And 3 is if there is any effect or change in the gut microbiota, it can be seen in the brain function. Or if there is any change in the brain function, it can be seen in the gut microbiota. Now, for instance, if we take Alzheimer's patients, their gut microbiome is different from the healthy patients, as in they have bacteria producing butyrate in less amounts and bacteria causing inflammatory response in more amounts. In the same way, today we've learned about autism spectrum disorder, right? So autism spectrum disorder children, those who are suffering from it, they are seen, they have a lot of gastrointestinal problems, digestion problems and constipation, etc. And because of COVID-19 pandemic, mental health has become such a global topic of awareness. So even in depression, the gut microbiome is actually different, and they have less amount of bacteroidetes and proteobacteria and more amount of firmicutes. So now let us see how prebiotics and probiotics are being used as therapeutic innovations.
Prebiotics are the non-digestible food fibers which help the host in maintaining its gut, and probiotics are live microorganisms which benefits the host. So, symbiotics is the combination of prebiotics and probiotics, and they help in modulation of immunal pathways, neural pathways, and metabolic pathways. So they help in reducing neurological disorders, inflammation, anxiety, depression, etc. And there is studies have proven that consumption of lactobacillus plantarum for 4 weeks has improved the autism associated symptoms, and consumption of consumption of bacterium brevi has improved the cognitive intervention in elderly people with memory complaints. And this is another therapeutic thing is the fecal microbiota transplantation. Collection of healthy microbiota from feces and transplanting it to colon recipient is a method. So I conclude with the therapeutic innovations, and keep your gut healthy and brain happy. Thank you.
Next we have Sparsh from Sri Sathya Sai Institute of Higher Learning, Anantapur. This will be an online presentation. Please maintain silence and listen to the presentation carefully. Next 2 presentations are online, so please maintain your composure and please listen to the work.
Once we start, you maintain
Hello, am I audible? Hello, am I audible?
We are not audible.
No, it's audible in the laptop.
Sir, आगे
Yes, ma'am. Am I audible now?
Depend minute, depend 5 minutes. We are maintaining 5 minutes.
Hello.
You are audible. And you are audible, but the time limit is only for 5 minutes. No? Please conclude this on the time. So you can start the presentation.
Okay, good afternoon everyone. My name is Parsh. I am from Sri Sathya Sai Institute of Higher Learning. a research scholar from the Department of Food and Nutritional Sciences, working under the supervision of Dr. Eastman. The topic of my presentation today is a study on the efficacy of probiotics in improving iron absorption. This is the research topic that I'm working on, and I'm studying the importance of probiotics in iron absorption. So you may have a question as to why iron, because iron deficiency is the most commonly observed nutritional deficiency in the world and also the most common cause of anemia. About 75% of Indians are anemic, and even WHO has been working in the reduction of anemia globally. From the literature reviews, we can see that iron plays a very important role in several physiological functions like cellular respiration and also genetic expression. The human microbiota, which is called the newly discovered organ, and iron is essential for its energy acquisition. When there is a deficiency of iron in the body, the translocation of intestinal bacteria is seen and the diversity of gut bacteria reduces. In the physiological form, iron is present in two states, that is the ferric and the ferrous, but it can only be absorbed in the body when it is in ferrous state. As you can see this picture, for the reduction from the ferric to the ferrous, there is an enzyme called the DCYTB, duodenoscytochrome B, which helps in the reduction from the ferric to the ferrous iron. So the probiotics act as an enhancer of this DCYTB enzyme, which catalyzes the reduction of ferric ions to ferrous iron and thereby improving the absorption. We can also see that many studies have reported an elevation in various iron biomarkers like serum iron, ferritin, hemoglobin, hematocrit, STFR and total binding capacity (TIBC). This is a short summary of the effect of probiotics on iron absorption, where dietary interventions are taken for clinical trials and the probiotic strains used for Bifidobacterium and mostly Lactobacillus species. Here we can see that the effect on iron absorption was there was an increase in the duodenal iron, an increase in the storage iron, ferritin, increase in serum iron and hemoglobin, and various other factors. A fruit drink containing probiotics with the strain Lactobacillus plantarum 299B showed an increase in 50% of non-heme iron. So, after all this literature, what gap we found was that there was no study that provided detailed insights into the precise molecular interactions of probiotics in iron metabolism. There was clinical trials which showed that probiotics are important in improving the iron absorption, but there were no mechanistic studies as such. Therefore, we decided to study and conduct an in silico study to understand the ferric reducing properties of metabolites of Lactobacillus species. This we did using molecular docking. Molecular docking is a computational approach in which we try to understand the interaction between two molecules. It can be protein-protein interaction or protein-ligand interaction. In our case, we have taken the protein, like I said, 2-deodeoxycytochrome B, and the ligands are the various metabolites that have been produced by Lactobacillus species. And after molecular docking, we went on to do the molecular dynamic simulation study, and the results show that out of the 7 compounds, 7 showed very good binding affinity and were selected further for MD simulations. For MD simulations also, the results revealed that all 7 ligands form stable complexes. After experimental validation, we can deduce that these probiotic metabolites could be potential enhancers for DCYTB protein. Therefore, further studies to understand the mechanisms of metabolites with respect to iron absorption can be done. This is just a brief result that I am showing. These are one of the 7 ligands which we have taken: hydrophenolactic acid, ferulic acid, and hydrophenoic acid. And this is the 2D and the 3D structures. This is the protein, and this is the ligand which is binding to the protein. In conclusion, I would like to say that recognizing the ability of probiotics to serve as facilitators of iron absorption is a pivotal strategy for the reduction of anemia. Therefore, iron-rich probiotic food formulations should be the focus of future research, since present studies, although few in number, have exhibited very promising outcomes. Thank you.
Next is Kirtana Sevi. Apurva, Kirtana and group. Apurva and group, Srivani Women's PG College, Malakpet. Please present your e-poster. Apurva and group from Srivani Women's PG College. Your time limit is 5 minutes. Please present your studies. Please put on your video.
Excuse me, can you put on your video, please?
These the complexity and interconnectedness of the factors that develop the process. This protective barrier for immune layer are in the precision and the immune. Both volume and special plant microbiome interactions under a single cell. There are three factors to consider that because particle reactor. Climate change can alter the behavior of both the host and the pathogen. It can also affect the interaction between host and the pathogen and can facilitate the emergence of new pathogens. Thus, plantain resistance plants. For example, production of extracellular polymerase substances, which is so this opens protected from the plants to prevent diseases, or they can produce stimulate plant growth. The third one is plant substitution. There is a system in place between plants and the microorganisms. This is in the right, the plants may rise under low iron conditions and increase the plant growth. You have an external body and evolutionary response.
Last 1 minute, ma'am. Apurva ma'am, can you hear me? Last 1 minute.
In conclusion, manipulating the plant cell microbiome
Okay, ma'am. Time is over, ma'am. Thank you.
technology will help in predicting the impact of climate change on plants and microbes and local environment like this.
Okay, ma'am. Thank you, ma'am. Thank you.
I request all students to sit in the hall. In another 5 minutes, we'll start the valedictory. Okay?
सर इसमें तो 2 लोग नहीं है सर एकदम ऑनेस्ट मैम मैम ఒక లిక్విడ్ జాయిన్ చేయాలి జూమ్ సెకండ్ పేజ్ కానీ ఇప్పుడు ఇదే మ్యామ్ జూమ్ अभी ज्वाइन है ना इसलिए नहीं हो पा रहे
अभी ज्वाइन
अभी तक नहीं थे मैं जब मैम बता रही कि मैम नहीं थे अभी आ गया ना
तो बहुत मुश्किल है।
सर अभी
मैम मना करती ना।
हाँ
टाइम हो गया 4:30 ये है ना कंक्लूजन तो 4:30 हो
हाँ
जाएगा।
सर हो जाएगा ना YouTube Lord.
अरे लाइव आ रहा है। ये इसके अच्छा अभी ये डाला ये कैमरा जितनी बड़ा नहीं कर सकते हैं सब पिन का पिन का हो जाएगा ये तुम्हारा है ना ये मेरा कैमरा है अभी उसको लगा दूँ क्या ये कैमरा
सर मैं अभी ये लगाया दोनों तो नहीं आएंगे प्रेजेंट क्या इधर पिन कर पिन कर दिया सर लाइव में तो जा रहा है वैलिडिटी के समय हम I know. You go to a shower sometimes. Then थोड़े से चलती हमें क्या चूसना नहीं साइड है लेकिन पता नहीं वो टेबल में आ गया सर ये
हमारा अच्छा चलता है अच्छा है हिंदी वाले से पहले भी हो जाता
Hello students, please maintain silence. In 5 minutes we start the valedictory. And I appreciate and thank all for your cooperation and interest, huge response, whatever you have given for this. And I hope you all had a nice experience last 2 days. So just have some more patience. We'll wait patiently so that our results will be out and you can go smilingly.
चैनल तो बहुत देर लगाएंगे थोड़ा लाइव
Yes, sir. Someone have lost the key. Just check your keys, okay? You have forgotten to check. It is on the table here. Keys. आ नहीं रहा है वो अगर से बुलाना तो इससे क्या ऑनलाइन नहीं किया माइक से भी ट्राई किया हाँ सर फिर इसके अंदर से बात हाँ तब लैपटॉप से चलो एक दिन के बाद అర్జున్ పనిచేస్తుంది ఓకే పనిచేస్తుంది ఈ టేప్ రికార్డ్ చేసేసి
ठीक जाइए
అక్కడే జరగాలి Mike.
And I welcome.
and my dear students to this guided session program. I would like to request Dr. Chaudhary, BBC, to visit us. I also invite Student National Seminar, Department of Microbiology, J. Sharada, convener, Student National Seminar, expert Dr. Sandeepta and Dr. Amita, stage. We are very happy to have Professor Sandeepta, Head Department of Microbiology, Osmania University, among us. She is our chief guest for the valedictory program. I am also happy to announce that we also have Dr. Amita B, Associate Department of Microbiology, Osmania University, who is also guest of honor for today's valedictory program. Now, I would like to invite Dr. K. Anuradha to present a report on the two-day national seminar on the microbiome evolution from fundamental science to therapeutic innovations. Principal, Dr. Chaudhary, Dr. Vivekananda College, Chief Guest of this valedictory function, Professor Sandeepta, Head Department of Microbiology, Usmania University, and also Director, CFRD, Usmania University, Dr. Amit Abi, Associate Professor, Department of Microbiology, Usmania University, co-conveners of the seminar, Dr. J. Sharda, Assistant Professor, Department of Microbiology, Bhavans Vivekananda College, Dr. Anju Nair, co-convener of the seminar, Assistant Professor from Department of Microbiology, Bhavans Vivekananda College. And we have distinguished professors, Dr. Professor Prakash Halami sir and Professor Deshmukh sir along with us for this valedictory function, and faculty of other colleges and faculty members of Bhavans Vivekananda College, organizing members of the college, organizing committee of this national seminar are here, and students of other colleges and students of Bhavans Vivekananda College. I welcome you all for this valedictory function. I'm sure all of you are eagerly waiting to know who got first prize for posters, who got first prize for oral and all. So I'm sure it is quite common to have that enthusiasm among the students. Before that, I would like to present the two-day report on this national seminar. It's my privilege as the convener of this national seminar and presenting this report. So the two-day national seminar on the microbiome revolution from fundamental science to therapeutic innovations is organized by Department of Microbiology under DBT Star College scheme in collaboration with Microbiologist Society of India on 24th and 25th January 2024. This seminar explored the scientific discoveries in the upcoming and mysteries in the field of microbiome. First areas like advancements in microbial-based therapies, precision healthcare, immunomodulation, and clinical research were focused. The topics included human, animal microbiome, probiotics, prebiotics, and postbiotics, plant microbiome interactions, environmental microbiology, bioinformatics and computational biology, and microbial therapeutics. 6 invited speakers participated in this 2-day national seminar. The first day of the seminar comprised 3 lectures. Dr. B. Shashikaran, former director, ICMR NIN, Hyderabad, spoke on gut microbiome, a vital organ for human health. Subsequently, in the next technical session, Dr. Jayanti Neelam Raju, Head Scientific Affairs, Unique Biotech from Hyderabad, she spoke on revitalizing gut health, unveiling the role of probiotics in restoring dysbiosis. Professor Bhima and Professor Veerabhadrachari, Professor Bhima from the Principal of Nizam College, sent an abstract on probiotics, a friendly microbial flora for enrichment of intestinal microbiome. Professor Brahmachari, the Registrar of Krishna University, sent an abstract for a oral lecture. You are what you eat, reshaping our microbiomes by changing our diets. Both of them being positioned in the highest designations because of their administrative constraints. They could not deliver the lecture. As a substitute, our colleagues, Dr. Pramita from the Chemistry Department and Dr. Shani spoke on the spoke on the efforts production as a prebiotic for postbiotic productions. The first day we also had oral presentations based on the theme of the seminar. So 6 research scholars presented their research papers in this slot. This second day of this national seminar started with the lecture, The Microbiome Revolution, Vedic Bharat. This was delivered by Dr. Deshmukh, President, Microbiologist of India, Professor and Head Department of Microbiology, University of Hyderabad. Thank you, sir. And the next technical session was given by Dr. Prakash M. Halami, Chief Scientist and Professor in CSIR, Department of Microbiology, Communication Technology, CSIR, CFPRI, Mysore. Sir spoke on microbiome formulation, CSIR, CFPRI initiatives. So these are the lectures given by the eminent speakers. We are very fortunate and we are really thankful to all the speakers who enlightened us in the field of microbiome. So along with the technical sessions, we also designed events for students. It includes research paper presentations and review of literature-based poster presentations. As I mentioned, 6 participants on day 1 presented their research work and e-poster presentation based on literature survey work. 36 participants are there in this theme and they have presented posters. The original research work on the theme of the seminar, that is microbiome, prebiotics, postbiotics, prebiotics, various posters were presented. So in this national seminar, 14 colleges took active participation. It includes the Development Department, Osmania University, Department of Microbiology, University College Government, Delhi, Andhra University, Rajasthan, Vidyapit, American University, Yedupur Delhi College, Brahmin Delhi College Government, St. Mary's College, Sri Satya Sai Institute, Srivani Women's PG College, Delhi College of Arts and Science, Manikla Verma Sharma JD College, Sharma JD College, University College Government, Delhi. So these are the colleges participated for this national seminar. This is the report for this national seminar, and we are really fortunate that when we have actually designed this seminar, our advisory board members of this seminar who actually suggested this theme. It includes the advisory research members of this seminar, includes Professor Gopal Reddy, retired professor from Department of Microbiology, Usmania University, Professor Geeta Sharma, Dr. Jyotirmayi from CFTR, retired chief scientist from CFTR Hyderabad, Professor Sandeepta, Dr. Amita. They are all our research advisory board members who have actually helped us in making this seminar into this shape. And I also thank our two resource persons who made this event possible. It includes resource persons, Dr. Srinivas Nayak sir from CPMB, Usmania University, assistant professor from CPMB, and Dr. Madhuri from Government Degree College for Women, Begumpet, who are our resource persons. Professor Geeta Sharma, she judged the posters for oral presentations. So I'm thankful to all the judges, invited speakers, resource persons, and the chief guest, and our University Star College team, who actually made us to think about this national seminar. Because we included a proposal, the probiotics, microbiome as a theme in Star DBT College scheme. And I'm really thankful to all the distinguished scientists who all are working in this area. We particularly picked up who all are working in this area, and based on the schedule, they have accepted our invitation, and we are really thankful for that. And not last but not least, without the support of Bhavans Vivekananda College management, our vice chairman, Air Commodore Jailan Shastri sir, and our dynamic researcher, dynamic principal, Dr. Chaudhary, who constantly support every activity that we propose forward, and he says that you go ahead with the activity. So for that, we are really thankful to you, sir. And I thank MB Society, DBT Star College team, Bhavans Vivekananda College, and all of our sponsors who made this event possible. Thank you all.
Thank you very much, Anuradha ma'am, for the elaborated report on the today national seminar. I would like to welcome Mr. Tanish to Professor Sandeepta with our Green greetings to ma'am, to our beautiful Sandeepta ma'am. Thank you so much for being here, ma'am. I welcome Ms. Shreya to present a plant hamper to Hamida ma'am. Ma'am works with plant microbiome, and a plant hamper is gifted to ma'am today. I welcome Niharika to present a plant hamper to our beloved principal. Along with microbiome of the plant is presented to principal. Let me welcome Mahesh to present a plant hamper to Dr. Kiranada, convener of the national seminar. Greetings and smiles for you, ma'am, for all your cooperation. I welcome Nazmi to present a plant hamper to our national seminar. Microbiology, senior-most faculty who have been one of the backbone for our activities. Thank you so much, ma'am. I would like to welcome our Dr. Chaudhary to present his views on this national seminar. Please, sir.
Check whether you are addicted or not. Is it really okay? I think I just spoke 1 hour back, ma'am, to all of you. No? Is it 2 days? Yeah, because I didn't feel it is 2 days. It was just 1 hour, 1.5 hour back I spoke to you. That is how the time moves fast, right? So first of all, let me thank Professor Sandeepta ma'am, Professor Amira ma'am for gracing this occasion and accepting our invitation. Before I proceed further, just I want some clarification. This is today national seminar. In this room there are many participants. There are all professors and no. I want to just see all the participants are happy with national seminar or not. Happy? Yes. How many? How much? So now, this is the confirmation that the national seminar was wonderful. Okay. A 2 days national seminar maybe once. Now this is not the standard of microbiome, it will increase now. I think we will not be satisfied with 2 days national seminars, and it should be a little bit more. And I thank all resource persons personally because they have enriched my students' knowledge. So it's my duty that anybody who enriches my students' knowledge, I should be the first person to thank. Because you're all happy, I'm also happy. The purpose of having this national seminar, I think we have achieved the objectives. Such eminent personalities coming and speaking to you, you will not get such kind of chance. Right? Maybe certainly when I did MS, I never heard about national seminar. Forget about it. I didn't even attend, forget about it. I didn't even hear national seminar when I did my MS physics. Right? And I think whatever little research I do in the college, I keep telling every guest that my mentor is Annaruga. Because I saw closely what is the research, how should we do it. Because I joined the college with only MSc, I didn't have any kind of research experience. But then I saw one department which is rich of experience in the research. Then I learned, when I associated with few faculty members, I think Shalini ma'am and Aparna ma'am, we work and we know. So before I become principal, I was very closely associated with the department. I have personally learned a lot of things from this department. And I think you will all be also learning and hope with the whatever one of learning you get from here, one day you will be standing like this speaking or sitting like this as a chief guest. Because Professor Sandeep Pranam is also our college alumni, being under this team of lecturers. Right? So that is what they give the outcomes. So I will not take much time. I will thank once again everybody. And I personally thank all the student organizers who yesterday and today for running around to organize this event. You made this event a success. You made every participant happy. So I thank all the organizing team completely from my heart. Thank you.
Thank you so much, Dr. Chaudhary sir. Let me invite Dr. Hamida B, Associate Professor, Department of Microbiology, Osmania University, guest of honor for this valedictory session to speak a few words about this program. I welcome you, ma'am.
Good evening, all of you. Principal Chaudhary sir, President of the Society MSI, Dr. Deshmukh, Chief Scientist, Dr. Helami, CFTRI, my colleague and friend at the Department of Microbiology, Osmania University, Head of the Department, Director CFRD, and Dr. Anuradha ma'am, HOD Bhavans, and Dr. Sharda ma'am, and all the faculty members of Bhavans as well as from the neighboring colleges, or I can say like all our extended family. We don't call more as affiliated college. We say they are our extended family from the mother department. That is what we got the input from our Honorable Vice Chancellor, and we all are under one family. And I would say, like they were saying, I just heard the last words of Dr. Chaudhary sir, that sir learned a lot from Anuradha ma'am. maybe it is something with the microbes and the microbiology, sir, because it is where, we get a lot of, motivation and input to do the things. And all of you are well nourished with the talks, right? Yeah, I think it's difficult for me to give a valedictory address. If you ask me to speak on microbiome, I can speak more. Yeah, I think there is some gut connection somewhere with the microbes, right? Yeah, it's nice to see all of you at the valedictory with so many smiles. Well, as said, you might have well-nourished talks. And I just want to add a couple of things to the younger generation, okay? From two concepts. That is, the microbiome research looks very, fancy, fantastic, and we want to do more. But at the same time, I also want to say that there is a lot of, not only the information is needed, but we do need a lot of sophistication when we want to do the microbiome work. And it's not only that you do the technical experiments, you also, a lot of big data is generated when you do the microbiome work. So I would suggest that how actively you participated in the 2-day national seminar, you also take up certain workshops or whichever, conferences or specifically the trainings which are based on metagenomics and big data management, because that is where it actually throws light on how you as a researcher or as a scientist can work on the microbiome. So this is from the technical part I want to say. And from the take-home message, I think morning you might have had a appreciated lecture by Dr. Deshmukh. See, being a traditional country and north to south, east to west, our India has diversified traditional fermented foods. Okay, so it is again a microbiologist only knows where is lactic acid bacteria sitting, where is yeast sitting, where is lactobacillus plantarum. Even if you say somebody it is idli, it is dosa, they will only look as a food and as a this thing or for the breakfast. But we, the ones who know what curd has, what milk has. So you people have to see that tomorrow if you want to go ahead in this subject from the microbiome concept, because I think most of the challenges which are unanswered, maybe microbiome science or the microbiome sciences will surely have the answer, the climate change and also the, because the recent pandemic, if you see a lot of reports on post-pandemic, you see the gut microbiota, how it was changed in the people who are having COVID-19 infection and who are having immunity to COVID-19 disease. So there is a lot in each, and you take arthritis, rheumatoid arthritis, you take cancer, colorectal cancer, we have seen some presentations of the students. So everything, the research is related to the microbiome. There is a lot shown, maybe the fecal microbiota transplant or the transplantation from the wild varieties in the plant system, many more new, because we may have the biomolecules based on the microbiome research. So there is a lot surely to do, but you have to be competent enough in the subject as well as the technical front. So these two things I want to tell to the students, focus on most, and I just was reading the abstract. I found one nice caption of the title, Harmony in Diversity. All of you agree, right? There is harmony in diversity. So I would say that maintain harmony among yourself so that we can make better India. And also I would say make harmony in your diet you are taking so that you have a good microbiota, and then you will be achieving the success or the wonders whatever you want to do. Good luck and all the best for all of you. Thank you. Thank you organizers for giving me this opportunity to come here.
Thank you, ma'am. We understood that our students have a lot of opportunities to travel, and it's only how much effort you put to travel in the field of microbiome. Now it's my pleasure to invite Professor Sandeepta, who is the head Department of Microbiology, Osmania University. She's also the director for Central Facilities for Research and Development. I stress on it, she's the director for Central Facilities for Research and Development, Osmania University, to share her views with us. I welcome you, ma'am.
On the dais and also in the audience, my fellow professors from Osmania University and also neighboring universities and institutions, and also the staff who is more like a family at Governance. As you all know, I have done my BSc here. So I'm also like a regular visitor to this college, and it's great to see my teachers. Dr. Ananda, who used to teach us when studying here. And I feel proud to come here always and remember the wonderful days when we were studying. So it's a very nice experience whenever I come here. And I'm sure some of you have attended the internship programs conducted by us at CFRD at Usmania University also. So, of course, I don't work in this area of microbiome, but I do read up because every time there is something latest which comes, we must be on par with it as a science person, isn't it? You're all biologists now. And I always tell that being a microbiologist today is being on top of our game, because post-pandemic, everyone depends on microbiologists now. You know, for the kind of microbiome we want to have in our body, to the vaccination, right? To epidemiology of the pandemic. I don't know if you have seen the movie Contagion. I always tell my students, go and watch Contagion movie if you want to know about corona pandemic, right? So I think everywhere you go, microbiology has a renewed importance now. So I was just reading on microbiomes, and it seems that top 10 startups on microbiome are working on how to engineer the gut microbiome without using genetic engineering. Okay, and each company has a turnover of millions of dollars, and the most interesting thing is some are established just 2 to 3 years back. Okay, so I think that as a student of microbiology, if you want to work in this area, you have a very good opportunity, because in India especially there are not many startups which work on microbiome. Maybe you see that ad on Instagram on Good Bug. You know, have you seen that ad, the Good Bug, and they challenge to say that they will change your entire metabolism. So I don't know how true that is, but definitely there is a lot of scope, because thousands of bacteria are there in your gut, all over your body. In fact, the number of bacteria is equal to the number of your body cells. I think you know that, right? So there is a wonderful scope in this area. So studying this subject is really, really important, because your microbiome is the way you behave. You know, your behavior depends on your microbiome. Your thoughts depend on your microbiome. So it's a universe in itself in your gut. It also tells you what kind of diseases you are prone to, right? So I think it's a very, very beautiful and still largely unexplored area, which can be definitely explored by people who want to contribute to society, especially as we are learning that several cancers are also depending on dysbiosis of your gut microbiome. You know what is dysbiosis, I'm sure, in the last 2 days of this conference, and of course what do you say? It's not prologue. Is it prologue, I should say? Yeah, it is prologue when Dr. Hamida has spoken to you about the microbiome. So it's a wonderful opportunity, definitely in science today. And I really commend the management and staff, and especially my teachers in the Department of Microbiology, who have taken this great initiative to make you understand such a wonderful latest topic, in science. And I hope that it inspires you to shape your career, maybe a startup in future. So our idea is always that teachers should have their students who are much more successful than them, which I always tell in whatever meeting we go. So I hope that you are all successful in your careers and choose a great field like microbiome in your career. And thanks again, madam, for inviting us to take part in this conference. Although I feel very awkward to say that I'm being invited as chief guest to my own college, I feel more like I'm a part of this occasion. So I also started like you in your place where you are sitting. I was also there, although I think at that time this building was not there. It was extended after a few years we left. 1999 was the year I left the college and joined Usmania University. So time has flown like anything. So you will find the same thing after the 10-15 years will just fly and you won't even realize how it has passed, how the time has passed. So please make use of this time and take guidance from great teachers like ours here. And we are also there in campus for any guidance and any training program. If you want any kind of training, please come to us and suggest that, ma'am, please can you do this kind of, want cell culture training or we want some training in Purbetter or HPLC, please come to us and suggest, make suggestions so that we will design a program for you. And since it is a state-sponsored university, we do not have very high registration charges. They are quite nominal, so anyone can afford them and get trained. And we usually get scientists from across Hyderabad, from central institutions also. So it will be a good experience for you also, and we'll be really happy to host you at CFRD. So you're all welcome, and I think that's all I have to say. Thanks a lot to the organizers, and all the best to the students here. Thank you.
Thank you so much, ma'am. Entrepreneurship is one buzzword and microbiome is another buzzword. It's good. I think microbiology students are having opportunity to connect these two terms. Thank you for reminding our students on this opportunity that they have in future. Thank you so much, Samvita. Pleasant evening, all of you. Excellent evening, very pleasant evening. But I know that there is some tension, some excitement in the hearts of the participants. Let us start the most exciting event of today's valedictory session. That's the prize distribution announcement of the winners. Shall we go to the prize distribution session? Yes, all of you. Yes. So, to bring up, we have conducted competitions in three areas. The first one is research-based presentation. The second category is research-based presentations. The third category is based poster presentations, where we have participation from faculty, research scholars, undergraduate students, and also postgraduate students. Now, I would like to request our dignitaries to give away the prizes. I welcome our guest of honor, our chief guest for the valedictory program, and I welcome other dignitaries on the dais to join for the prize distribution ceremony. I would love to thank our esteemed judges, Professor retired professor of Osmania University, Dr. A. Harti, assistant professor, and also Srinivas Nayak, CPMB Osmania University. In the first category, that is research-based oral presentations, I call upon Sindura, research scholar of BBC. She presented evaluation of antibiotic potential of floral ancient plant extracts. Congratulations, Indira. I would like to announce that Pushpa Jain have won her second prize, but she presented online, so she's not among us. She won the topic comparative study on the impact of commercial fungicides. Pushpa Jain, I hope you are watching the live streaming of this valedictory session. Congratulations, Pushpa Jain, for all the hard work. You have won second prize in the category whatever you have presented. Congratulations, everyone.
Thank Thank you. Thank you very much, ma'am.
Moving on to research-based posters. I would like to welcome Alanka Ketan Kumar and Umashri from AV College of Arts, Science and Commerce. Hearty congratulations to Ketan Kumar and Umashri. Second prize in research-based poster presentation goes to V. Vaishnavi Lakshmi and K. Swati, again from AV College of Arts, Science and Commerce. Congratulations, Vaishnavi Lakshmi and Swati. We have a very huge participation in the category of literature-based posters. I would like to invite Professor Halami Garu and Professor A.M. Deshmukh Garu to join the dignitaries for giving away the prizes. Thank you so much for joining for the prize distribution, sir. In the category of post-graduation students, literature-based posters, the third prize goes to the topic modulation of gut microbiota by bacteriocins presented by Divya. Congratulations, Divya. She is from BBC. She presented on the topic modulation of gut microbiota by bacteriocins. Second prize in this category goes to Alekhya, Aishwarya Reddy and Pranati. Alekhya, Aishwarya Reddy and Pranati of Bhavans Vivekananda College. Alekhya and Pranati. They presented on the top unveiling the potential of probiotics in the relief of lactose intolerance. And the first prize goes to the duo team Alankrita and Harshita. Students of Microbiology Department, Bhavans Vivekananda College. They spoke on the topic, the skin microbiome, current landscape and future opportunities. We also had a huge participation from undergraduate students where we are announcing 2 prizes for UG students. The second prize under UG category goes to Sadbhavana, Nihal and Gayatri Sharma. Receiving second prize. The spoke on the topic communication between microbiome and first prize under UG category goes to the power of probiotics, presented by Nikki Kumari and Anshul Singh. Congratulations, Nikki Kumari and Anshul Singh. It's our pleasure to give a memorable memento to Professor Sandita. I would like to request Dr. K. Anuradha and our beloved principal. Yes, congratulations to all the prize winners, and I would like to thank all the dignitaries for giving away the prizes. Thank you so much. I request Principal Sir and Anuradha Madam to give away a memorable memento to Professor Sandita. Chief guest for today's valedictory session. I also request our Principal Sir to give a memento to our guest of honor.
ये तो लाइव से मंगा लो ना होगा मंगा लेंगे तो बोलो कि यार वो लाइव वाला पेज ही चाहिए अपने वाला पेज तो क्या अपडेटेड चीज
अरे ट्राई करेंगे
है ना तो वो
I would like to request Madhuri ma'am to come on to the dais to receive a memento. Madhuri ma'am was the judge for our poster session. She is the assistant professor from Begumpet Women's College. I request all the dignitaries to join the audience. Now we have prize distribution for students from Microbiology Society of India. For this session, I would like to hand over the mic to Mr. Mahesh for further proceedings.
Good evening to all the respected dignitaries who are present here and my fellow friends. The Department of Microbiology, in collaboration with the Microbiology Society of India, is organized an exhibition on January on the occasion of exhibition on microbial products. The three teams have been won prizes. Due to some reasons, we are unable to give the prizes. So today we are announcing. And I request Deshmukh sir along with the principal sir to give away the prizes. The first prize is given to the Liquid 3 model. The participants, the winners are Avantika Bhunda and Naga Sushma from BSc MGC third year.
Using a place. Yes.
The second prize is won by the MSc Microbiology students on exhibit of exomicrobiology. The students are C. Sirisha, Nikita, S. Vidya, Kaveri, Harshavardhani, and Sharon Perana. textbook product of Vedic Microbiology, again by MSc Microbiology students. The participants are Rajesh, G. Shreya, P. Divya, Bhumika, Shilpa, and Manaswini from BSc second year.
सर इनको ही तो देखना है ना हम देख के क्या
The Department of Microbiology and our collaboration with Microbiology Society of India as a part of HIV awareness, we've conducted international infographic competition from throughout the India. There are two prizes won by students. The first prize goes to Radha Prasanna and G. Manasri. Second prize goes to from MSc Microbiology students Torit Gubya and V. Shilpa. The Microbiome Society of India, as a part of encouraging the students to participate in various things, every year they give the Best Student Award. The students are based on so many criteria, based on their participation and their activeness in the programs. From the MSc, the Microbiome Society have selected Tanvi as a Best Student Award. Thank you, sir.
Thank you all the dignitaries for your kind prize distribution. I welcome Dr. J. Sharda, convener of the 2-day national seminar. So we have calendar distribution, calendar presentation from MB Society of India, Microbiologist Society of India. I welcome Dr. A. Madhavi and Professor Deshmukh sir to present calendar for the year 2024. So as Deshmukh sir mentioned in the morning, the only microbiology calendar in the world. So we have calendar 2024 presentation to Professor Samhita Calendar 2024 year presentation to Dr. Hamida Bin. I request Hamida Bin madam to receive a special calendar, microbiology calendar. We receive many calendars in the new year, but this is something special for a microbiologist. Very thankful to Microbiology Society of India for presenting such nice calendars. Request her to present vote of thanks for the two-day national seminar. My greetings to the dignitaries of the guests, now they are in the audience, distinguished guests and all the delegates. I think we are coming to the end of the program. On behalf of the organizing committee, I stand before you with immense gratitude, and it is indeed a proud privilege for me to propose this vote of thanks. A sincere thanks to Chairman Sri D. Prabhakar Rao for his constant support and encouragement for conducting this national seminar. Vice Chairman A. Tamil Sri Jain Shastri VSM, BVB Sainik Puri Kendra for gracing inaugural session and preceding the inaugural session. We extend our heartfelt thanks to our esteemed chief guest for the inaugural Professor B. Sashikiran, former Director, National Institute of Nutrition, ICMR, who has honored us with his wisdom, experience, and valuable insights. His presence has elevated the stature of the national seminar, and we are truly privileged to have served for the seminar. We express our deep sense of gratitude to our guest of honor for the inaugural, Professor Geeta Sharma, retired professor from Usmania University, who is our teacher, guide, and mentor, for accepting our request and enlightening us with her valuable inputs. We express our sincere thanks to Rear Admiral Sanjay Dutt, VSM, Commandant, and other officers at CDM for their kind help in providing this wonderful auditorium and also for providing infrastructural and logistic support. I thank the Principal Sir, Dr. G.S.B.R.K. Chowdhury, Bhavans Vivekananda College for his constant support and encouragement at every point of time in organizing this event and making it a grand success. It wouldn't have been possible without Sir's support and participation. We could collaborate with MD Society with the help of Professor K.M. Deshmukh, and I also thank BBT and the Star College scheme as we got a financial support for this program. We express our sincere thanks to all the invited speakers. Dr. Jayanti Raju, Head of Scientific Affairs, Union Biotech, Professor Bhima, Principal Nizam College, Dr. V. Brahmachari, Dean and Associate Professor, Department of Biotechnology, Krishna University, Dr. A. Deshmukh, President of NPK Society, Dr. Prakash M. Halami from CFTRI, who have accepted our invitation gracefully. Your support has been instrumental, and we are thankful for their collaborative effort that has gone into organizing this national seminar successfully. We thank our judges, Dr. A. Madhuri, Coordinator of MBA Society for the entire state, and Dr. P. Srinivas Naik from CPMP for accepting our invitation and judging various competitions held during this program. We extend our thanks to print media for their presence and coverage of this event. We thank Chief Guest
for valedictory session, Professor B. Sandeepta, Head Department of Microbiology, Usmania University, and also Director of CFRD, and guest of honor, Dr. Hamida B., Associate Professor, Department of Microbiology, Usmania University, for gracing the valedictory session. Your presence has elevated the stature of this event. We express our special thanks to the members of the committee of this national seminar for their constant support and untiring efforts, who have worked smilingly under stress and strain to make this national seminar a grand success. We thank Mr. Shriver, Office Superintendent, and all other supporting staff of our college for their kind help and constant support in arrangements. We thank active participation of faculty members of other colleges, research scholars, students. Your enthusiasm and active participation are the driving forces behind the success of this event. We hope that the knowledge shared here today will resonate you and contribute to your professional growth. Our sincere thanks also goes to the organizing committee, volunteers especially, all those who are behind the scenes who have worked tirelessly to ensure the smooth execution of this event. Your dedication and hard work is tremendous, and we are always be remembered your hard work. Last but not least, our gratitude also extends to the audience. Your attentive presence and engagement have added the vibrancy to this gathering. We hope you will continue to be part of the upcoming sessions and contribute to the success of the national seminar. As Professor Deshmukh sir has announced, we are looking forward for an international seminar also. So I expect the same response even for our events ahead. So wishing all of you all the best, and thank you all once again. I request all to stand for the national anthem.
Request participants to collect their certificates at the entrance door. Except MSc Microbiology students, everyone can collect their participation certificate. I request all MBSI Society students to please stay back. Microbiology Society of India students. Fine.
Students and staff, one group